Okay, well, thank you. I know everybody's probably getting hungry and ready to go on a boat, so I'll try to make this as informative as I can, and obviously we'll be on the boat if there's questions. I'm happy to be talking to everybody. So I guess a couple things. Obviously, I want to thank DebRA. It is an honor, as everybody else had said, to be able to come and meet here and meet people that I've had a lot of indirect contact with over the years. The interesting part is I would say that this is probably the most exciting conference I've had a chance to come to, but it's also the most nervous for me. And I'll explain that, and then I want to go into what we've done so far. Excitement-wise, like I said, I've been involved in this active ingredient for probably over 10 years. And it's been refined, reformulated, higher concentrations have been made. I've always felt that this active has the most potential to treat the skin effects that you see with EB. And I think one of the things that Dr. Bruckner mentioned this morning, which I thought was very, very good, is that this is a disease of the whole body. Obviously, our therapy, what we're looking at is treating the skin effects. The nice part, which I see with some of these other talks, is people are also looking at trying to develop therapies that can treat other things than the skin. So there's an opportunity as new things come up, you have different types of therapies that can be combined. So to me, it's very, very exciting that all these things are going. The one thing I will tell you with a lot of the research that's been done has been on the skin. There's been probably over a dozen therapeutics that have been tried in the skin in EB, been published trials. These are randomized trials, blinded, so you had a placebo. Nobody really knew what they were on, and nothing has worked. One of the things Dr. Paller, who will be here on Saturday, she'll be talking about our first study that she actually conducted. This was done completely at her facility at Northwestern without any influence from anybody whatsoever. And the data that she got, which I'll talk about today, had to do with a breakthrough therapy designation that we got with EB. And again, I'll demonstrate and talk to you about the importance of that. So that's my excitement. I hope to get a chance to meet people more personally while we're here. And again, like I said, it's an honor. The one thing that makes me nervous, and I've done talks, probably hundreds of them, and I can honestly say I really have never been nervous at a talk, is I feel such an obligation when I see everybody out here in the audience. My job here is we have a therapy obviously we believe in, but the question is, can we test it appropriately to get it over to the hurdle? And if it is work, that the FDA accepts what we do, that it's rigorous enough, and as Dr. Bruckner mentioned, which is one of the key things, is having assessments that are objective. That's really the key when you talk to the regulators. You can have a drug that you believe in, you can have a drug that you can run all these different things, but they have to be able to see that black and white, you have a drug that works. And that's one of the difficult things when you do clinical trials, and I'll talk about that a little bit, is because sometimes you do things that are not in the norm that you would do when you treat your patient. Because you're doing certain types of measurements that can show a difference that you can feel comfortable that it's not biased and it's real. So that's kind of where I feel very nervous, because every day the reason I get up and my team is here, we're focusing on getting this therapy tested appropriately. That's our goal. We can believe in our baby as much as we want, but I want to make sure that we move this forward as quickly as possible, test it as rigorously as possible, and if it comes out as we hope, it's the sooner we get it out there. It's one of the first therapies. So that's what I live for each day. So let's go to the next slide here. One of the things I do need to step back at, and I do apologize, when we set up to come to this meeting several months ago, we had started our clinical trial in January with the hope of enrolling actually a little bit quicker than what we did. We enrolled in 3 months, which is very quick. We were hoping to do it in about a month, and to have the efficacy data here to present. Unfortunately, we don't, and I'll explain to you why we don't and when we will be presenting it, and hopefully it'll be soon. So just a little bit about SIAderm. So we're a small company in North Carolina. Just to give you an idea, we have 7 full-time people, a very, very small company. One of my co-founders is here. We'll talk about it. This company was actually formed about a year ago, and I'll show you some of the timelines that we've done in the last year, which are actually pretty quick compared to industry standards. We've actually been working on this program, like I said, for several years, and we self-funded it ourselves. We believed in this program. We tried to get it to a point where we can try to get additional funding. Once we get into the clinic, the pricing and cost gets very much more than what we put into it. So that's kind of what our goal was, and that's where we are. So it's a topical therapy. We're looking to develop this across all different EB subtypes. So you'll see in the demographic slide on our study that we just finished, we had patients both in simplex, we had recessive dystrophic, and we also had junctional. Now, one of the things that Dr. Paller will talk about on, whoops, this thing just went out, on Saturday is back about in 2010, 2011, Dr. Paller ran a study at her facility. It was an open-label study, so these were all patients that were given an active therapy and had some very, very promising early results. And she'll be talking about those and going over some of those results on Saturday. One of the things that was a benefit from there, the FDA put out a regulation a few years ago called Breakthrough Therapy Designation, which is a designation that is products that have very compelling early clinical data in a high unmet need area that basically gives you almost unprecedented access to the Food and Drug Administration. We were fortunate enough with that data that Dr. Paller generated to be the first biotech to get that designation. Now, one of the things I will tell you, it's a blessing, and in some cases it's a little bit of a curse, but in most cases it's a blessing. The blessing is we've had unprecedented access with the FDA. It's been much more of a collaborative process. We've had multiple interactions over the last year where on average when you're developing a drug you may have one every several years. The downside, which I say a little bit is not as positive, is that since it is so collaborative, all along the way you need to be collaborating with them. So as data comes out and things change, they're the first people of entry that we go and have to talk to them about the data. So that's why we can't put the data out as quickly, because they're the ones that we really need to go through and talk to as we move forward. So just a little thing to give you a perspective on what we've done and what clinical drug development or drug development is. So before a product goes into clinical trials, whether you're going into healthy volunteers or actually patients of the subtype, there's a whole lot of work that needs to be done. And my partner and I actually did all this work, funded it ourselves over a several-year period. That involves not only getting all the manufacturing in place, you have to make sure that you've got a product that's safe, not only from a point here where we're going to be testing in the patient, but in this case, make sure that it's not going to cause any undue bacterial contamination, other things. So all that was done by ourselves. And then you have to do a whole level of animal testing. That's required by the Food and Drug Administration and also regulators all over the world. So when you go into the clinic, every day that you test, you have the same number of days in animals that show that that product, at least in animals, is safe. So all this work had to be done over a several-year period. Last year, we opened what's called an IND. That's basically it's called an investigational new drug application. It allows you to go into the clinic, okay? And that was done, again, last January. And then I'll talk about where we went. There's phase one. Phase one is typically healthy volunteers. Usually the Food and Drug Administration wants you to test different doses and make sure that your product's safe before you go into the patient population that you're going to go to. So we did a phase one study, and again, I'll go through the timelines. We've completed a phase 2-3 study, so we're right here. And go on to the next slide here, just to give you a perspective on how long do those pieces take. So pre-IND can take several years, and basically that's what it did with us. But when you get into the clinic, if you're going basically from what I would say the IND stage, which is right here, the first in man, up to here, on average is about 4 to 5 years, maybe 6 years, depending on the situation. Okay? And you can see, and then plus then it could take another 3 to 4 years to get to what you would call a filing to the FDA. So just superimposing it on what we've done over the last year. So we opened the IND, which is right here, in January of last year. And from January of last year to now, which is about 16, 17 months later, we're right here. So we've done a lot of work and shaved off several years. So it just gives you a perspective of how dedicated we are to get to this stage where we want to be able to go and talk to the FDA with our data. So that's kind of where we're at this point. So just a few things. Again, we've had these discussions with investigators, but just to give people in the room here a perspective, like I said, you have to do very, very extensive animal testing. And you have to do it across all types of species. That's a requirement regardless of what the pharmaceutical is that you've got to do. So we have to do it in dermal studies. The animal of choice is pigs. Their skin is as close to us as any other species. So we have to do studies in pigs, we have to do studies in other animals, including monkeys, we have to do them in small animals and so forth. But the key point here is what we've seen, and we have concentrations of SD101. We've tested it at 3% with a Dr. Paller's study. We've tested it 3 and 6%, with 6%, which I think is the optimal dose for our product moving forward. And we've even tested at doses above. We haven't seen any safety issues of concern. So, and just to let you know, in the animal studies, we've done treatment up through 9 months. So these are what's considered chronic animal testing. So we've tested it very thoroughly, because the way I look at it is you're putting a product into a patient population that you've got to make sure, at least with all the testings that you can do, that it's safe. And we've done that. The other thing I will tell you here, we've had discussions with the Food and Drug Administration. We've finished all of our animal testing. We've also reached agreement on several other key things on our program in terms of safety, and also one of the next steps we'll be doing after I show you a little bit on our phase 2B study is our next meeting with the Food and Drug Administration is what is the trial that we need to do to demonstrate if this product is ability to get registered. And that's, again, is our goal. We obviously believe in it. That's what we get up for every day. But whether the rigors are going to be met, we hope it will, and we're going to do everything we can to make sure. So I want to just talk about at least what I could share with you now on the 2B study. So this study, just to let you know, is probably one of the largest randomized placebo-controlled trials that's been done in EB. And what that means is people did not know whether they were on a matching placebo cream, whether they were on the 3% cream or they were on the 6% cream. And that's the same that goes with the investigators. So nobody knew, and we didn't know either. So it's a completely blinded. Everybody is blinded until the study is over. And so we had 48 patients. We had them divided into one of 3 arms. And again, that's done electronically. Nobody knows who gets put into each arm. It was topical application of the product once a day for a period of 90 days. Now, again, looking at objective responses. So from the Food and Drug and even the European regulators, the most objective response in a disease where you have wounds and so forth is wound closure. So that was our primary response. We picked lesions that had a certain character, our wounds in this case, that had a certain characteristic. They had to have been open for at least 3 weeks. They had to be a certain size. That was our primary endpoint. We used cameras and we used very objective equipment to be able to take photographs before and after, measure the wounds, and so forth. Again, agreement with the regulators that this is an objective endpoint. Now, we looked at also several other types of assessments, which I would call more subjective, but also just as important. One of them was looking at the whole body coverage. How does the body coverage and lesional skin change over time? Now, again, that's a little bit more subjective because for some people to say what percentage is covered versus others, there is variability, but also very, very important. We also looked at a couple other things, and I think Dr. Paller will mention it in our open label study on Saturday. We also looked at any change in itching and pain. In the study that was done at Dr. Paller's site, and again, it was an open label study, there's suggestions of some improvement in itching and also in pain, which again are very, very important in the whole picture of the skin effects that you see with this disease or this disorder. Another thing that we looked at too, and hopefully Dr. Paller will mention that information, is we also, the Food and Drug Administration actually asked us to look at how did these wounds look after they healed? Did they scar? And we also had that information, and I think she'll share that with you on Saturday. And those are also things that we looked at in the study here that we just completed. And obviously on the bottom, we monitor safety. In our trial, we monitor safety basically daily because that is paramount. The results as far as efficacy, we won't know till the end, but safety, we want to make sure we're not putting anybody at risk, even if it is a blinded study. So this is just the demographics of the study. So these are basically the patients that we enrolled. So like I mentioned, we had 48 patients. We had 11 that were simplex. We had 29, I'm sorry, this is mixed up. It's actually switched. We had 29 recessive dystrophic and 8 with junctional. The average age was 12 years old, ranged anywhere from 6 months up to 44 years. Equal distribution of males and females in our study. And as far as the primary endpoint, which again is the objective, the average lesion size or the wound size was 14 centimeters squared, and you can see the range. A couple things what I can tell you on the study is we monitored safety throughout the study, and we saw absolutely no safety signals of concern. And again, this was in a blinded fashion, but it was very important because one of the things we were able to do with this study is allow people to roll over to open label of the 6% SD101 after the trial was over. So we wanted to make sure before we got to that point that we were monitoring safety on a daily basis to make sure there was nothing that stood out that we needed to either go in and potentially unblind the study or go in and look and delay rolling people over. So again, we did not see anything. So what wound up happening is we had 44 patients that completed the study. That's not unusual, that you have some people that either discontinue or actually they just are not able to participate because of, it could be personal reasons, it could be financial reasons. Although, you know, they don't pay for anything in the study, but sometimes even just to travel and so forth can be very difficult. So the good thing here, we had 44 patients that completed the study, all but 2 rolled over into the open label study. So everybody rolled over, the last patient rolled over the end of June, so we have some patients that are on as much as 3 months. And we're going to monitor, people will be staying on that open label study up until either if we find out that our product, when we test it into a larger study, doesn't work, or we get the product registered. So that's the situation we have here with this particular program. So just next steps, and again, I apologize, you know, I was hoping to have some of the data. We obviously, we just got the data recently, some of it this week. We need to analyze it, it takes time. We need to go down and talk with the regulators to design our next study. And hopefully that study will be the one that will, if it's positive, will allow this product to be out there and available. So our next steps, again, right now we're analyzing the data to optimize, as I showed you in that slide before, the phase 3 registrational study. That's our next step. We plan to hopefully meet with the regulators soon, give them our results. We have a protocol for the phase 3 study. We may be able to optimize it a little bit based on the results of this study. It depends on how we analyze it, or leave it the way it is. And that's something we're going to do. And we're hoping very soon to initiate this phase 3 study. It'll be a study likely that'll be both in the US and in Europe. And hopefully we'll have some new people, because people that are currently on the open label will not be eligible to participate in this because they're on open label. So that's kind of where we're at. I thank you for your time. I wish we had more data to share, but we will be soon. So thank you very much.